Hello. Thank you, Oxford Nanopore, for the invitation to present our work to you, all of our community. I'll talk about some of the new developments and recent ones in DNA and RNA sequencing that our group has been testing and share some early results from those evaluations. First, let's talk about R10.4.1 ultra-long DNA sequencing. This has been around in the field for about a year. We have been piloting this from early access up until recently and been very impressed with the performance of the kit, and the throughput has improved over time. More recently, we consistently are able to get over 100 gigabases per Promethean flow cell with read N50s for these experiments ranging upwards of 100 kb. For the uninitiated, ultra long really means read length of 100,000 bases or longer. So for our 10.4.1 ultra long DNA sequencing experiments, we typically get an accuracy of around 98%, which is very impressive. In addition, what this allows us to do is very high quality, state-of-the-art single nucleotide variant calling. And what you are seeing here is a table that talks about variant calling numbers using Pepper Deep Variant that was developed in collaboration between our group, UC Santa Cruz, and Google Health. And the numbers that you see are corresponding with HG002, a genome in a bottle sample, which is used for benchmarking. And you'll see numbers for indels, which is standing for insertions and deletions, and single nucleotide variants. There's a lot of numbers. The take-home numbers that you care about in terms of just a summarized version of it is the F1 score, which is giving you how good the performance is. What you'll notice is that for single nucleotide variants, SNVs, the performance with Pepper Margin Deep Variant is 0.998. This is state-of-the-art. It's better than what short reads give in the field. And this is the kind of performance that allows us to do Stanford university-based ultra-rapid sequencing project type applications, where this pipeline is what we have used in this work, and being able to do very high quality variant calling. As you'll also notice, the indels have a little bit of a low F1 score, and that needs to be improved, which you'll see an update on in the next slide. In addition to this, we've also been working with Duplex DNA sequencing chemistry, also for a number of months now, and that has improved quite a bit too. More recently, we consistently get over 60% of the reads that we achieve from a Promethean experiment as duplex pairs, which is impressive. And that means half of those, so 30% of the experiment is typically on the order of duplex data. We anticipate that this number will improve as the London Calling Conference proceeds and we see an update from the Oxford Nanopore team. What's also been impressive to us is the accuracy of these duplex data, which in our hands for median stands at around 99.7%. We are working with the ONT base calling team to resolve why we don't see the Q30 median performance that they see in-house. But nonetheless, this is still very impressive performance on the accuracy. This allows us to do even better variant calling if we wanted to do it. And with Pepper Deep Variant, again, if we look at the SNV performance, it's better than already better 1D simplex F1 score. And the indels also show a documented improvement from 0.88 to 0.92 on the F1 score using the same HG002 benchmark data. What's also interesting to note is that if we break down the indels into the repetitive regions versus the non-repetitive regions of the genome, then in the conventional regions of the genome, the performance on indels is just about as good as it is for single nucleotide variants. In the repetitive region, it needs improvement, and it's something that various groups are working on improving. The combination of ultra-long and duplex also allows us to do very high quality assemblies, and that's some work that we have underway in collaboration with other groups like the University of Nottingham, and we'll share updates over those as time goes. Now, another very recent early access that has come into the field is the RNA004 kit. And this has been a fantastic and long time in the making update. For a longest time, the direct RNA sequencing accuracy was based on the RNA002 kit that's available in the field. We started using this in early access in about 2016. And for a good few years, the accuracy of a platform with Nanopore direct RNA sequencing was around 86% on the median. A combination of software updates made this increase to 89.8% in around 2021 with the release of an improved higher accuracy Guppy model. What's also worth noting here is that these two graphs that you're seeing on the screen are coming from the same data that were rebase called with the newer update that came in 2021. And this was an impressive improvement to go from 86% on the median to nearly 90%. But what was a significant improvement was this. With the RNA004 kit that released essentially about a month ago in early access to a handful of laboratories, including ours, we documented the median accuracy of the data at 95.6%. This is a remarkable improvement, and as I'll show in the next few slides, it's an improvement not just on the numbers, but also in the power of the performance of the technology itself. And what I mean by that is let's try and break it down by different categories of RNA that we can sequence with Nanopore right now, and especially using this RNA004 kit. Everyone, as we know, will sequence poly-A-RNA with direct RNA sequencing. That's something we've done for a few years, including many people in the community. And with the new kit, we can consistently get 4 to 8 million reads using as little as 100 nanograms of poly-A-RNA or 1 microgram of total RNA. This is quite nice. The read N50s for good preparation of RNA material range between 1.3 to 1.6 kb, which is quite nice. And this is based on human poly-A-RNA. In addition to this, we can also sequence ribosomal RNA. There are a handful of manuscripts that have been in the field that have sequenced ribosomal RNA directly using Nanopore platform over the last few years. With the new kit, we can sequence human ribosomal RNA as well on a Nanopore. The figure that you see here is showing a histogram of human ribosomal RNAs. The peak on the left is corresponding with 5S and 5.8S ribosomal RNA. The peak in the middle is correspondent with 18S ribosomal RNA. And the peak on the right, it's actually more like a blip. It's just that very tiny blue colored blip right below where it says 28S corresponds to 28S ribosomal RNA. Now, why we don't have as many reads of 28S as compared to 18S? Is the issue based on abundance, adaptation, or something else? It's a question that we are in the process of resolving, and we will have some resolution and improvement on this over the ensuing few weeks. In addition to ribosomal RNA, we can also sequence human tRNA on a Nanopore. We've done this experiment with the RNA004 kit as well. There's again a handful of manuscripts that have sequenced tRNA directly on a Nanopore, and this work goes back from foundational work in the 2015 to 2017 time, and then over the last few years, multiple groups have published studies analyzing tRNA directly using a Nanopore. And we can see tRNA reads from human RNA on a Promethean using the RNA004 kit. And not just that, what's impressive is that we can do all three of these simultaneously on a single flow cell. So our most recent direct RNA experiment on the Promethean used human poly-A-RNA, human ribosomal RNA, and human tRNA all adapted together and sequenced simultaneously on a single Promethean flow cell. And we got a reasonable amount of data for each of these categories. And this is both exciting and very powerful because this opens a whole range of applications, because imagine being able to do this from a microgram or two of starting total RNA material. And that's both a significantly lower input requirement and also a significantly improved output in terms of the throughput that you'll get and the inference you can make. To give you an idea of what this kind of data processing can do and what the improvement is, here's an example. What you're seeing here are two panels. The top panel is poly-A-RNA reads aligned to the mitochondrial reference chromosome in the human genome. These poly-A reads are from our 2019 study where we had sequenced GM12878 poly-A-RNA. And what you'll see in this IGV plot that's shown for visualization is a reasonable coverage of the entire mitochondrion, the coding RNA and the polycistronic strands. What you'll also notice is that the gray boxes have a lot of red colored or multicolored stripes. These multicolored lines essentially represent base level mismatches. So this is a base mismatch error in the data. And what this is showing is that there was a significant amount of mismatch error rate in the data at the time, which is fair. The accuracy at the time was 86%. Now, if you look at a similar plot on the lower panel, these are poly-A-RNA reads coming from the UHRR material, which is a mixture of 10 cancer cell lines, total RNA. And we look at poly-A-RNA aligned to the mitochondrial chromosome We see a similar pattern as to the top, but now you'll notice that these colored lines have significantly reduced, which is essentially saying that the base level mismatch has substantially reduced on the newer data, which have 96% accuracy per read on the median. In addition to that, what you'll notice is that on the panel on the upper part, the slope on the coverage tends to be a little bit sharper, and on the bottom, it's not as sharp. And also, the blips that you would see in coverage because of the presence of homopolymeric sequences, so repeats of nucleotides 3, 4, 5 or longer, have significantly been improved in the newer chemistry in 2023 with RNA004. So in summary, combining the Nanopore ultralong and Duplex DNA and with this fantastic update on direct RNA using RNA004 has been very exciting. We have various assembly and transcriptome experiments underway, as have many other groups in the field. And we are excited that this combination will yield very high quality annotated complete genomes, both human and non-human. And this is a very exciting development for us as academicians working with the platform. With that, there's a lot of people to thank from acknowledging our group, various collaborators at Northeastern University or outside, as well as our various collaborators across different institutions. A lot of people at Oxford Nanopore who are our colleagues and collaborators. And thank you.